Yeah, good morning. We're so happy to be here, and we're gonna take you through kind of a normal day of the stuff that we do when we come out in the field. And first, let's introduce the team. I'm Becky Ostertag, University of Hawaii Hilo. I'm a professor there.
Hi, good morning, everyone. I'm Susan Cordell. I'm the director of the Institute of Forestry, which is the U.S. Forest Service in Hilo, and I'm a co-lead on the project with Becky.
Yep.
Hello, everyone. I met you folks already, but my name is Kupono, and I'm a member on the team.
How's it? I'm Sophia, and I'm also a member on the team.
Morning, everyone. I'm Kona. I'm the field leader. Met you already, but excited to have you guys follow.
Fantastic. And Maybelline, please introduce yourself.
Hi, everyone. I'm Maybelline. I'm also part of the crew at Pilina, and I'm here in the lab. You can't see it because I have my background on, but hopefully when I turn it off, you'll be able to see it.
Fantastic. Fantastic. Thank you so much, Maybelline. And thank you so much, Kumu and students, for joining us today. All right, so we're going to turn it over to Rebecca, and we are happy to accept your comments and questions in the chat. Maybelline will be gathering those, and we'll check in with her periodically to see if there are any questions and comments, and if so, we will answer them. Does that sound good?
Great.
Awesome.
All right, so we're going to talk about our project. This is the project that Susan and I and another colleague, Peter Vatusik from Stanford University, wrote a grant proposal for originally in 2011. And it's taken a lot of morphs and changes through then, lots of different aspects. But the idea behind this project was we had been working in these lowland wet forests in Keaukaha area on the Big Island for about 10 years at that point, and we had been seeing pretty much a lot of failure of our efforts. We tried to be restoring the native forest, getting the native forest to come back. You could see ohia trees and llama trees, kopiko trees, but they were really getting crowded out by these highly invasive species. And we tried to remove the invasive species, but guess what happens? They just came back. So, and that was like years of research that it took us to like find that out. And we did a lot of reading and a lot of hard work, and we realized that wasn't going to work. So this project was morphed into the Liko Nā Pilina project as a way to come up with another strategy to try to restore these lowland wet forests. Because these lowland wet forests are a really important part of our heritage, our biological heritage, our cultural heritage. And there's not much of them left. These would have been the most diverse, tallest, biggest, most impressive type of forests in Hawaii. And there's so little left due to agriculture, housing development, invasive species, climate change, all these different threats that have affected our landscape. So our idea was to make a new strategy of introducing introduced species that were non-native but non-invasive, as well as the natives, put them together in restoration teams to try to keep out the highly invasive species.
Yeah, and so for this project, you know, the thing about restoration that's so important is you have to have a goal. Like, why are you doing it? And, you know, for us, we had 3 goals in this project, and that was to increase native biodiversity, reduce invasion or increase invasion resistance from the species that we selected, and slow down the carbon and nutrient cycling. And we'll talk more about that later when we're out in the field. So we actually selected all of the species using a scientific process, and we used functional traits to select our species. And functional traits of a plant are kind of similar to like how we all differ as humans. You know, we're all different heights, sizes. We have different color hair. We have bigger noses or good eyes or bad eyes, all these different traits that we have. Plants have those too, and it defines how they work in the ecosystem. So we measured all of these traits of the plants and then sort of plotted it in an experimental fashion, did analysis to show which species would work well together, because you don't always want everyone the same. We need diversity to have a functioning forest. So we tried to select species that had different ways of utilizing the ecosystem, like they grew taller or they grow slower or they have big leaves or small leaves. So it's a way of sort of like fantasy football where you're getting all of the species to work together for a common goal.
And we're really amazingly grateful for our partners here at Keakaha Military Reservation, the Hawaii Army National Guard Environmental Office. So graciously, the field site is on their property, and this is their greenhouse, and they're very important partners in what we do, because this kind of strategy, testing it out here, is a test bed to see if this can work other places as well. But it's been successful, and it's been showing that we can make the forest into a better, more functioning forest that's not only good for the biodiversity, for the plants and animals, but it's also good for the humans, because it makes it a lot easier to walk through the forest, to do training through the forest, and to meet those kind of goals. And it also requires less maintenance in the long run. So we're really grateful to have these wonderful partners with Hawaii Army National Guard Environmental Office.
Yeah, I think the way you can define a project as successful is if people will utilize the way you chose these species and use it themselves for their land management needs. And they're doing that here at Keaukaha Military Reservation. So we're just really excited that they're happy with the project and they see the success, and we work hand in hand together.
And also what we're learning is, because it's working here in this environment, which is very similar to many other environments in Hilo and in East Hawaii throughout the Puna region, that this kind of technique can work in other places in Hawaii, and in a different way could work in other places in other ecosystems around the world with different kinds of species, but similar kind of strategic approach in choosing species. So that's a background on the project. Did we leave anything crucial out?
I don't think so.
Okay, so our typical day, we're going to take you through a typical day. So our typical day starts here at the greenhouse, and Kupono, Sophia, and Kona would come here first thing and kind of get started. Do you want to explain what you do here? And we'll walk around the greenhouse a little bit.
So one of the first things we do every morning is make sure all the plants have been watered and they've been taken care of since we last left them. Because we do care for such different types of plants, we just want to make sure all of them are able to stay happy, because we do outplant these and we propagate them all the way from seed. So we become slowly attached to them and really want to make sure they survive when we outplant them. So we have a lot of different compositions of plants, and a lot of them are outplant species that you'll see again in the plots when they're more mature. This is alahe'e, which is also used as like a dye plant. And so a lot of our plants also have like more usages than just being a plant. They can also be used for different things. This is our kamani. He has a lot of a longer, thicker leaf than our alahe'e we just saw. And so you can start to see the traits of the plants even when they're at this really tiny stage. These have like little, almost berry-like fruits, and this one is a larger nut. A lot of people use kamani for oil. And then of course we have our noni, which a lot of people use the fruits as medicinal purpose as well. And the pigs like to eat them, so it helps feed the wanderers around the environment. We also do outplant things like mamaki. It's used as a medicine as well. A lot of people consume it as tea. A lot of people know chocolate tea, and that's made from mamaki. Even though it's part of the stinging nettle family, it really doesn't hurt. And then like all its fruits and its flowers actually happen on its branches instead of being out like you see on like mango trees and such like that. This is our kukui. A lot of people know kukui by when we use nuts for leis. It also has a medicinal purpose. And I think it's our state tree.
Yeah.
Yes. But then we also have things that were in this forest when we originally started, such like our kopiko. We have a lot of pre-existing trees of it in our plots, and a lot of our seeds from they do come from these already pre-existing kopiko that have been here for a long time. So we're just re-putting them back into the forest.
Maybe go to one other greenhouse and then we'll go to the field.
To here. Okay, well we are at the second greenhouse, Sophia.
Yes, so we have a total of 3. One we have where we start our propagations. The one we just came from is sort of like our more mature plants that will get outplanted, and this is like our teenager stage, where they get a little bit more exposed to the sun before we outplant them, because we don't want to shock them with too much sun at one time or too much heat at one time. So a lot of our slower growing plants are also inside this greenhouse, such as our ohia. They're very important to our environment. A lot of species depend on them and their flowers. And because of recent things such as rod, a lot of them are having a hard time in our forest, but we do try to outplant them as well. We're just extremely careful with them, you know, making sure they don't get any cuts or anything. And we really try to facilitate their growth, even if they're a little bit on the slow side.
What is rud? You mentioned rud.
So rod is Rapidohiada, and it's sort of like a fungus that likes to get into the plants, and then I think it clogs it up so it really can't have water and such, and then the plant ultimately dies from it.
Great. I think probably a good time to go out in the forest. Does anyone, this might be a good time to ask for questions.
Do we have any questions, Maybelline?
So they haven't written any questions in the chat, but let's go to Kumupili to see if they have any questions live.
We have a kind of general question. What are the biggest challenges you faced when you've been cultivating all of these plants? Like if there have been certain maybe plant sicknesses or complications you may have had when growing either a specific plant or all the plants, and if anything has been a problem and how you guys kind of overcame that or are still trying to overcome that.
I'm happy to tackle that question. So we grow tons of species out here, species that were historically present. But unfortunately, we can do excellent work here at the greenhouse, and we can get these to an adult stage ready to outplant in a great way. But as soon as they get out there, we notice that there's some pests. We have problems with pigs. We have problems with storm events. So the biggest issue that we have here at KMR is just outplanting and survivability. But here in the greenhouse, I mean, we have great success. We do a lot of research into proper propagation techniques. So we're pretty good at propagating the plants we have in house just when they get outside.
Anyone else like to add to that response?
I'll add one thing. Since many of the species that we chose for this project many of them are canoe plants that were brought over by the Hawaiians. And some of them are also fruit plants or agricultural species like mango and avocado. And we found that they get a lot of disease and pests more so than the native species. And so having to deal with agricultural species has been kind of an adaptive thing that we've had to deal with in this project.
Question before they get out and like come back on to the video. Does anybody remember the different types of plants that they were trying to outplant or trying to grow, propagate in the greenhouse? Give me like 3 or 4 plants, that's all.
One says kopiko, mana, kukui, and mamaki.
Awesome, guys.
Yeah. And before we get to the beginning of the research process, getting out into the field, we were gonna just quickly finish up our interview with Sophia. Sophia was telling us how she got interested in agroforestry and just falling in love with nature in general.
Yeah, so I started out here when I was doing my associates at Hawaiʻi Community College. My degree plan was tropical forest ecosystems and agroforestry management. And after I got out here, I noticed that I like field work a little bit too much some days. And so to be able to facilitate that, and so it can be something that I can do later on. Now I'm currently at Hilo doing an environmental science, bachelor's of science degree. And then hopefully after that, I can transition into doing more field work.
Fantastic. Thank you so much for sharing, Sophia. And so we'll happily take questions about what Sophia shared at any time in the chat, and we'll check back in with Maybelline for that in a little bit. All right, so now we are here with the team, and they are getting ready to head into the field. They're going to open with an oli.
Yep. And please feel, we're going to do eho mai. You're welcome to join us if you would like to. And we're getting a little bit of, well, right now, like that.
Eho mai ka ike mai no mai.
Oh, no, clear. Oh, no.
Okay, so now I'm following the team into the first plot.
one of our experimental plots. We have an experiment where we have 4 different treatments, and a treatment is a mix of species, 10 species, we're calling them teams. And so we have 4 different teams, and we're interested in those teams or treatments, not just how the individual species do, but because we do want to know that, like, does Kopiko grow better than Kamani, or we want to know all the differences and who fruits and flowers more and things like that. But we also want to know how they work together as a team. And so our treatments are 4 different teams of species. We're interested in which team does best at which things, which teams grow better, but especially which teams keep the weeds out the most, because that's really one of our key metrics, is to try to decrease the weeds in the amount of weeds and the amount of management that we have to do in this kind of forest. Want to add anything to that?
Yeah, we're also, of course, as I mentioned earlier, interested in increasing native biodiversity. That's one of our goals. So we also want to see how these teams work, because light environment, how much light comes to the forest, is one of the critical key things for native plants to be able to survive. That's one of the reasons why you don't see a lot of native species in like a forest of strawberry guava, because there's too much dark for the ohia and other species to survive and grow. So we're hoping that with our teams here, we've manipulated the light environment to be the right environment to promote native biodiversity. So that's also one of the key things that we're trying to look at here.
Fantastic.
You guys want to add anything?
Yeah, happy to. We're very fortunate to be able to work comparatively working in this plot type, which right now we're in a slow redundant plot type, which I don't know if Becky and Susan got into slow redundant.
We didn't get into that for them.
Yeah, so we're able to work in a much nicer, more navigatable forest type than the horribly invaded forest that's right around us. But like they mentioned, there's some incredible native species here. I just want to point out this guy. This is Hapu'u. Really, really important native tree fern. We absolutely love seeing it out here, and in this block especially, there's a lot of young Hapu'u seedlings that we're excited. So it's really convincing and reassuring to see the plots be successful when you start seeing these guys regenerating on their own.
Amazing. Amazing.
Okay, so now we're gonna walk to our next plot. And we're gonna get inside and do the typical measurements, show you the typical kind of measurements that we would do. So we'll walk there now.
Okay, so Maybelline, we're gonna transition to the next part. I'm gonna turn off my audio real quick while we walk. And thank you again for the nudges in the chat, letting me know about the audio. Really appreciate that. And thank you, students and Kumus. I'm so thrilled that you all could join us this morning as we're out here in this beautiful forest. Okay, be back shortly.
I guess she's keeping her video on so we can watch how they walk from one plot to the next. You can all see like they're walking through like rough terrain, considered rough terrain because it's all I hope I'm pronouncing that right. I didn't say, but I'm originally from Pohnpei, so Micronesia, so not from this island. And so pronouncing some of the Hawaiian words is really hard for me. You folks have a lot of H's in there. H's in my language only makes the sound longer. So like your kahaku, is that what makes the sound longer? Yeah. So actually pronouncing it, it's very difficult for me. And then saying words without them, like yeah. Yeah, so there's like a lot of that they're walking on in between plots, as well as other invasive species that they have to walk through.
We take a wellness check on the tree once per year. So each tree has a name or an individual tag number. This is 2733. And this is a roost or nene leao. And we are concerned with a few things. On our data sheets, we collect information on height. We also collect information on diameter of the tree. So we're concerned about the thickness of the tree from it's at breast height. So we get the measurement always to be consistent at 1.3 meters above ground, which is something that we're able to identify on our body using our height pole. We know where 1.3 meter is. So then we line it up with our cells and we find the spot on our body that assigns to 1.3. So we don't always have to quick measure. So that's one thing that we're concerned about. We also measure height, like I mentioned, using our height pole. This is a great arm workout. You get to run the pole in the air all the way until it hits the top of the canopy, and they click like this.
You
Here,
want to just measure this tree?
we can absolutely
just
go
do
for
it.
Yeah, let's go for the measurement on this. And we also get to see immediately, because we keep last year's data, we check to see how the height has improved. So, Kupono, what was the height for 2733 last year? This should be in quadrant D4, spot 8.
Yeah, I'm in 6. My bad. Hold on. What was the number again?
2733.
DBH.
Okay, I got it.
Okay, great. So yeah, how did he look last year?
7.16.
So yeah, while we're measuring height, one person, which in this case is myself, gets under the tree, but it's hard to estimate from underneath how close you are. So you usually have another observer in the team go out and tell you how it looks to the highest point of the canopy.
You gotta go higher.
A little bit higher.
Yeah.
Tell me when we're good. Feel good about that, Becky?
Yeah. Yeah. I think you're good.
Cool. Yeah. So last year was 7.16. Cool. So this year he actually did grow. So this guy is around 7.38 right now.
Oh,
Hi.
yes, it grew. That's wonderful.
Sure. Yeah. And then other things we're concerned about, besides also diameter, we're checking for fruiting and flowering of any of the species within our plots. So for roofs, their flowers are large yellow bundles that are very seasonal, which we're not really in season right now for the roofs, but we're getting close. I think if we would give it a few more months, we'd see a lot more roofs flowering. But for this individual, we would mark no fruiting or flowering currently.
You want to take the DBH?
What was that? What was that? That term?
Do the diameter at breast height, DBH.
So for me, about 1.3 is about here. And so you never want to be on like the very low side of the tree. So we always try to be consistent and be sort of on the topper side. And for here, because he is going to be more than 1 centimeter, this would have 2 stems. And so you just go to each stem and be like, when you do your tape measuring, you always do the smaller to larger.
I have no questions in the live from the kids, but maybe they have some live in the classroom. Let's go to Kumupili to see.
And if not, we'll just keep walking to the next spot or to our next spot. And if you have questions, feel free to drop those in the chat, and Maybelline can give us a heads up if we have any. Does that sound good, Maybelline? Thank you.
Can do that. can do. And if they don't have questions, I have some that I wrote down for them.
Fantastic, fantastic.
We'll go do the litter.
I could also just talk about one of the things when you do research is things don't always go as you expect. You write everything out on paper, perfect, but in the real world, things happen. And so we have to adapt. And that's just part of doing research. So a couple things in this spot to show how we've had to adapt. We talked about rapid ohia death earlier, and I don't know if you can see that, but there's a number of trees that have died from rapid ohia death in this plot. And, you know, we were counting on that canopy to be part of our experiment. So we have to adapt because it no longer is. And also, if you look behind you, there's a one of our plants here, challah, the pigs really love. And pigs came in and decimated. It had a search image for this particular species, and it just came in and decimated them. So we had to deal with a pig problem, you know, and so we're working again with our partners. They invested in putting a fence around this whole enclosure. So, you know, just want to show that, you know, you can't always just count on things, you know, working as we expect, and having these kinds of things come in really, it's important for us to keep our observation skills strong and learn how to adapt. Absolutely.
Okay. So another thing that we do every month is litterfall. So let's go to one of the litter traps. When a plant puts out a new leaf in the canopy, it's going to drop another leaf. So we can't really measure. It'd be really hard to like look up and count how many leaves are in the canopy, but we can measure how many fall down by putting these traps. And so that gives us an index of how much growing. And you can see there, anyone recognize any of the leaf species in here? Well, it looks like we have some mango, okay, and some ohia, and anything else in there?
I'd call this llama.
Yeah, llama. Okay, so we will collect what is in the trap, bring it back to the lab, and we dry it in a drying oven, and that's set at 70 degrees Celsius, and that gets all the water to the leaves, because the leaves, it's wet, right? And we want to get the dry weight. Everyone does the same method wherever they are in the world, because a leaf in Hawaii might weigh more in water than a leaf in Arizona. So we want all the leaves to be like uniformly dry before you weigh them, so that all scientists all over the world can compare.
Yeah, so another one of our annual surveys we do is seedling surveys, which we're looking at regeneration of all our outplant species. And for example, what we do is we run a mirror tape transect through the plot. For this instance, I just did one of the quadrats, which is we divide our plot into smaller subplots to help us manage and record our outplants, and especially the census. It helps finding and navigating to where the individuals are. But so yeah, for this example, we're in one of our smaller subplots within the plot, and we're going to look at seedling regeneration of our species. So we align it every other meter along the transect, just to make sure we're not doing any kind of observer bias, researcher bias, and picking where we want the quadrat to go. For this isn't an exact spot we would do it. Just wanted to process when we do have seedlings. It takes a long time to learn what the seedlings look like and how to distinguish them between the invasive plants, especially. But here on this log, this is what we see a lot of when we have these decomposed old ohia logs, moss blankets start to form on them, and then you just see an explosion of ohia seedlings. Really, we will count on some of these logs hundreds of ohia seedlings, which is great to see. We love seeing that kind of regeneration, but unfortunately, we understand maybe out of the 100 that we see in a very condensed area, 2 would make it up to adulthood. But of course, that's just the survival strategy they went with. They threw out thousands of seedlings, so it's still important information. And we also count the seedlings by size class. So we count seedlings based on, we have the measurements on the data sheet. I don't have them on the top of my head. I think Kupono will have it. It should be under the census data sheets. I don't think it's inside. I think it's under. But yeah, so we record them by size class, and also, yeah, Kupono will have the different sizes.
I was just trying to show what Pono was showing, the spread or the sheet that they fill out for seedlings, like how we fill it out. So just an example.
Perfect. Thank you, Maybelline. Thank you so much.
I guess one more thing to me real quick. The fun part about seedling is we get to monitor the same seedling individuals every year, and we can notice how much they're growing because we can track. And then as soon as a seedling gets up to that diameter at breast height level, it'll get a tag and it'll get recorded and added to our overall census list. So it's really exciting.
It becomes an adult.
It becomes an adult.
Oh.
So, this is an ohia that definitely qualifies now. He will get a tag the next time we do census and sapling. So yeah, that's just, I'm excited to see. We're getting a lot of regeneration of ohia here.
Do you folks in the classroom have any questions for the field crew today?
Don't see any questions.
Just give them a minute. I think Kumu Pili is talking to them right now.
Understood. Thanks so much, Maybelline. You've been absolutely amazing. Thank you so much.
She's gathering a lot of questions now. She's looking all over.
We do have one question about Nenelau, wondering if it grows like abundantly in the wild, or because we think we might have seen it, but we don't.
Yeah.
We're just wondering.
It grows, it's a native species, and it's kind of like a pioneer type species. It likes disturbances, so it'll grow like along the roadside. I see it a lot like on Hamakua coast, but I see it on Saddle and in Hilo too. Anywhere where there's been soil disturbance, you'll see it.
I have another. A Nene Leao is a resprouter, so it doesn't just rely on seed to grow.
An awesome question. Do you guys have more?
We have a couple questions. We've met everyone except, sorry, it cut off at the beginning for someone's name in
Susan.
the. Okay, I'm
Was
sorry.
it-
Can we
Is
have
it
your
Susan?
name?
I think
Susan?
was it Susan?
Yes. Okay, sorry, we really wanted to know know your name because it was cutting out, and we've met everyone on our field trip, so we haven't met you yet. So
Oh, okay. Yeah. Hi, I'm Susan Cordell. I'm a co-lead on the project with Becky. You just don't ever see me because unfortunately I sit in my office all day.
Oh.
But I my name is Susan Cordell, and I'm the director at the Institute of Pacific Island Forestry. We're the U.S. Forest Service.
Cool. Hopefully we get to see you on one of our field trips.
Yes, I look forward to it.
And then another question is a little bit more personal, but do you all have like a favorite part of doing all the work that you do? Because I feel like my favorite part would be like planting trees, but maybe all of you guys have something specific that you really enjoy doing.
Okay, who wants to start?
I know it sounds odd, but I really like weeding. It's very It's like meditation, and it's sort of like you're sitting around and chatting and weeding, and it's a good time for making observations and looking around to see how things are doing. So that's my favorite part.
I really like measuring the tree diameter. I like to see how much things have grown and who's lived and died over the last year, and just kind of getting a snapshot that way of the forest.
My favorite part is sapling. So when we finally get to graduate our plants into getting their tag and everything, because sometimes it can get a little depressing out here when you see certain plants die. And so seeing sapling is like the new growth and everything. So it's a very happy time.
Yeah, I can share about the lab. So I am currently in the Becky Ostertag's lab, where the Liko Nā Pilina brings in their samples to process. So I can do like a quick walkthrough to show you guys like the lab. So I'm like sitting at one of the benches. This is just a 360 view. And then I'm back, but as you can see here, we have our data sheets. This is where we come and we weigh the leaf litter. We have scales here. Sorry, this is awkward to carry around my laptop, but this is like our drying oven. Can you guys see that?
Yes, we can see that.
Yeah. So we come and place them in there in like paper bags. You don't want to put anything plastic in there, or it might melt. And you can set the temperature at the bottom. It's set to 70, and because I opened it now, it went down to 69. But yeah, that's our drying oven. Usually our leaf litter come in like these big paper bags, and once we have them dried and weighed, we come to this fume hood, and we have a grinder in there. And then we grind down like our soils, I mean, not our soils, grind down our leaf litter into like powdery little substances. And it ends up in one of these. So the end process comes out in like those little coin envelopes. That'll then be like sent out to be tested for whatever needs are. And so that's it. That's our lab.